So thank you very much for the introduction, and I want to thank the organizer for giving me the chance to share the work that I have done with the Thai group. So, I have been working a lot on case, disease association tests, focusing on the HLA test, and I have this imputation system that using the population-specific reference that can impute the HLA for different populations. And then since I'm in Thailand today, so I want to share with you this rather new results that we have. We actually, it's the first time we show this result. So this is a collaboration with Professor Natia and Professor Patarin that we are focusing on this SLE. So just a very brief introduction, that is a multi-organ autoimmune disorder that affect mainly the female of the childbearing age. And then it actually, since it's an autoimmune disease, it's actually produce a lot of antibody that attacks our own cells. And you can see from the picture there that a lot of the organs are under the attacks, and you can see all the different different symptoms that are shown by the patients. And so SLE is primarily affecting primarily the black, where we see that around 100 to 250 in cases among 100,000 persons. And the second most common one will be the Asian, which is around 90 to 100, and then followed by Hispanic and the white. And then talking with my collaborator, it seems like there are reports that the SLE cases in the Asian population is actually higher in severity, where they see that 50% of the patient actually will suffer from this infection, especially in the lower tract of the respiratory tract. So I did some digging of what has been studied for this Thai population, and Professor Tokunaga here has published this first paper about HLA and SLE back in the year of maybe 2002, so around 20 years ago, that they have identified that this DRB1502 and DQB10501 is susceptible to SLE. And then in recent years, I think 2 years back or 3 years back, that Professor Natia Lab has published this nice GWAS that focusing on the Thai SLE populations. And as you can see from the Manhattan plot there, that the major peak is still in the HLA regions, and then they identify several others novel SNPs that is not being reported before. But since today our discussion is on the HLA, so I will be talking about this HLA association. But a little bit more digging. So what have been known about this HLA association in all the different populations? As we know that some alleles may share between different populations, but some of them may not. So we want to look at the pattern of this association. And then we look at two-field resolution. And then we actually found that if we group them into the serotype, the DR2 and DR3, in this case, that you can see that a lot of them share the same risk allele. Well, in this sense, you can see that the Thai is carrying the DR2 alleles, which is carrying in nearby populations such as the Chinese and the Japanese are reported to be carrying this the same susceptible DR2 as well. And DR3 is more specific to the European, as what we can observe from the table here. And beside DR2 and DR3, of course, there are some others that are reported others, but they are not replicated in other populations, so we won't discuss too much about that. And the table just now was talking about susceptibility. So this table is trying to looking at the protective allele instead. So you can see that protective allele-wise, the DR11, DR13, and the DR4 is reported repeatedly by different populations. And again, in this case, we see that Thai have this DR12, and then the other population like DR11 are mainly the European and the Indians, and DR13 as well. So those are the backgrounds. And then next, moving to our methods. So again, same as the last two presenter, we are using the next generation system as well, but then we are not using nanopore or PacBio. We are using the EON S5. So you can see all the three different platforms here. So we are using the all-type kit from 1 lambda, and we have sequenced the Thai sample. But in this case, we are not actually directly sequencing the Thai SLE sample. We actually, with collaboration with Dr. Surakarma and Professor Tokunaga, that we have sequenced around 400 Thai healthy individual. And then so the process of determining is that we are using the GeneDx NGS software to analyze the result. And since the kit is from 1 lambda, so we are using their default software as well for analyzing the result. And then finally, is the main point of this presentation, that so using those raw data, we have tried to build a Thai population specific imputation reference. So the concept of that is that I will go briefly on that, is that so for training the data, the imputation reference, we need two kinds of data. So first we will need the NGS type data or any HLA typing data which is good enough accuracy, and then we will need a SNP microarray data. In this case, we are using the Affymetrix Axiom ASA array. So by using these two data, we put into our software, the HiBack software, and then we will come up with a software, with a so-called model, where we call it the parameter estimate. And then with this parameter estimate here, what we can do is that, for example, we have the SLE sample here. What we do is we match the SNP in the Thai SLE with the SNP in our model, and then using our prediction software, we will give what is the best predicted HLA alleles there. So again, I want to emphasize that this is not a direct genotyping. This is a prediction method that we can so-called predict the HLA allele by only looking at the SNP microarray data. Of course, I understand that some of you might start worrying about the accuracy, but don't worry, I will show you what have we achieved in the later slide. Going a little bit more technical stuff is that how we actually build the system and what kind of SNP that we are actually using to build this system is that the beauty of this system is that we found that we cannot use the SNP inside the HLA gene. It's simply because a lot of selections and recombination is happening in the HLA gene itself, so that actually the tagging power of the SNP inside the gene itself is rather weak. So what we come out with a solution is that we are using the SNP outside of the HLA gene, and those SNP are actually quite relatively stable. And then because in the revolutionary sense, they are there for a very long time, and then so they have the ability to tag the SNP, the alleles inside the gene itself. So by saying that, what we do is that we are using the bootstrapping method, that we randomly choose the individual from our training set, the 400 sample, and then at the same time we are randomly choosing the SNP from the surrounding region of the HLA gene. And then what we do is that, so this is our iteration that, so for example, at first you can see that we choose a random number of SNPs, and then you can see that our first imputation accuracy result is around 50%, 47%, not very good. So what we do is we add more SNP, and if the SNP doesn't increase the accuracy, we remove them. And then we will do this. Of course, we are not doing this manually. We are using programming to do that. And then, so you can see that by doing that, we can increase the SNP, the imputation accuracy from 47, and slowly we will reach a plateau where you can see that we can reach a plateau around 94%. But again, some of you, the bioinformatics guy in the room here, you will be arguing. So the first selection of SNP will be very crucial then. if the first selection of SNP is very bad, no matter how you add the SNP, it will still be bad. So what we do is we repeat this process 500 times. So again, second time, this one is better model. This is 96% now. And then again, we try again, but this is 95, so we are not using this, we are using the 96. So we repeat this 500 times to choose the so-called the best tag SNPs that can impute all the different HLA alleles in our model. So as you can imagine, HLA alleles can be very polymorphic, and then within the 400 sample, the maximum is that we have to predict 800 types if all of them are heterozygous. Of course, some alleles are more common than the others. But then one of the questions I always being asked is that, so how many SNP that you actually need to impute? Because some of you might think, then I just genotype this SNP, then I can predict the HLA types. But unfortunately, the SNP that we need is quite big. We need around 300 to 400 SNPs. As again, why we need so many SNP is that first, we need the SNP to tag the different allele. And secondly, we have the SNP that's saying that this is not the allele as well. So we have the yes and no SNPs. So that's turned out to be quite a big number here. And this is the SNP for each gene. So this is HLA A genes, and then HLA B have another set of SNPs. C have another set of SNPs. So after we build our imputation system, what we do is that we are matching the SNP that we have finalized with whatever the SNP genotyping array data that we have. And then by doing that, we so-called predict what is the best genotypes. So we are predicting them in a haplotype form. So you can see that we predict both of them together, and then this is so-called the posterior probability. So you can see that in this case, there are two possibilities of the final answer, but the bottom one show a higher posterior probability. How is this probability calculated is that we are looking at how well the SNP that we have and the target data set. The better the matching, the higher the score. So in the end, we will have so-called this kind of table that's showing all what is the sample 1, LE1 and LE2, and the probability. And then we actually did a simulation study, what is the cutoff that we should use for throwing away all the badly imputed SNP, badly imputed samples. So, and then you can see that this is so-called the summary of the accuracy distribution. So for this case, you can see that if we cut at 0.5, we are actually could save around 90-something% of the sample. So we are only failing around 4% of the sample. So it's a 96% success rate of the imputing. So next is that, of course, accuracy is the most crucial point. So we try to do simulation again with our newly built imputation panel. And we have a 2-field imputation panel and a 3-field imputation panel. And I will explain why we need 2 of them. So first of all, let's look at the 2-field resolution that you can see that at the last column here, we are reaching around 98 to 99% of imputation accuracy. So again, we are not 100%, we are missing around 1 to 2%. So, but for research use, I guess that is good enough because you just need a 5% difference to show significancy, and we are only missing 2% of that. And then, but if you compare this 2-field resolution accuracy with the 3-field resolution accuracy, you can see that 3-field resolution actually dropped a bit. But as you can imagine, and as what has been presented by the 2 presenters just now, you can see divergence when you look at 2-field and 3-field. Maybe originally there is 1 allele only at the 2-field, and then when you look at 3-field, it becomes 3 alleles. And in order to build our reference, we need enough sample that carrying that particular alleles so that we can build the model. So as you can imagine, if the sample size stay the same and you are looking at higher resolution, of course, the accuracy will drop. So the only way to solve this is to increase the sample size. So again, we are imputing for this 8 HLA gene at the 2-field and 3-field resolution. sorry, I'm taking too much time on this. So let's look at the result. So again, this Thai SLE sample is collected from 2 hospitals in Thailand, from the Chulalongkorn Hospital and the Ramatipoti Hospital. And so we did the HLA imputation, and we do a QC of the badly imputed SNP, and then we do a case control association test. But I don't have the time to go through all of them. But so this is the top 3 associated alleles. Yeah. So as listed here, and then I want to let you know that this is replicate what has been published before by Professor Tokunaga group. But again, we are increasing the resolution to a 3-field resolution, and also this is the top 3 protective allele, and why I highlight them together, as you can imagine, they are in the haplotypes. So again, this is also concordance with previous publication. So beside this top 2 association, which is already being reported, except that we have higher resolution, we actually see that we have others alleles that are also associated here. Like nobody has studied about DQA1 before, but as you can imagine, most likely they are because they are pulled by the DQB1 and so on. So we will have more detailed analysis that whether they are in the same haplotype or not. So we perform the haplotype analysis. Again, I don't have time to go through this, but I have tried to summarize it in the graph form so that it's easier for me to discuss this. So maybe this is a rather new way of showing the HLA haplotypes, that the bar heights of the height of each of the bar represent the HLA frequency in the Thai SLE patients, and then the gray line that connecting them, the gray line that connecting them is showing that whether this particular allele is in the same haplotype with the other allele or not. So you can see that, for example, if you focus in on this particular allele, you can see that all of the people who carry this carry the other alleles and so on. So I run out of time, but then I will just show you what we have done. So we have done the logistic regression to condition on that, like this is the major haplotype that we have, and then we have a new alleles that is never been reported, the DPA1 alleles that is being split into like half-half, and then we perform a logistic regression and show that they are actually weak linkage between this haplotype and the DPA1. And then lastly, so those are, replicate the previous result, and then we want to look at the subphenotypes of the SLE, and luckily that the Thai group actually collected a very intensive subphenotypes of these Thai SLE patients, and then you can see that the percentage showing there is that how many of these patients is actually suffering from the particular symptom that is listed there. Cutting the story short is that we did a case control analysis on all the subphenotypes, and then you can see that there are 2 highlights there. We have a malar rash and the discoid rash, and both of them are rashes. And we found that they are both associated with HLA. There are no other symptoms that is associated with SLE besides these 2 different rashes. So again, I don't have time to go into the detail, but you can see that this main association is in DQA1, and then because when you look at DQA1, you always look at DQB1 haplotypes, and then we found that actually it's the DQA1 alone is the significant ones here. And the malar rash as well, you can see that the DPB1 is the significant one, but when we look at haplotypes, we actually found that the haplotypes is actually a better representation of the malar rash. And also the same alleles has been reported for a similar disease called the systemic sclerosis. So it actually brings some meaning that we actually see the same HLA association. And this is a summary slide that, so we started by a GVAS, and then in order to maximize the benefits of the GVAS, so we built an imputation system and then performed an association study and found that these 2 new association with rashes. Yep, thank you very much.
Thank you very much for Dr. Charles and for the excellent talk. and deeply in reporting in 6 digits of the HLA allele with HLA. Now it's time for question.
Hi, so my question is regarding the autoimmune diseases. So when we have autoimmune diseases, sometimes we have, multiple, two diseases or 3 diseases together. So in your cohort, did you find any other, associated autoimmune diseases besides SLE? Did you try to see in your cohort?
So let me clarify the question. So you're asking whether I can see that the association that I found is also associated with other autoimmune disease?
Yes.
Yeah, well, first of all, our data do not have the information of other autoimmune disease. So we don't have real life data to see that, at least in our data set. But then some of the alleles that being reported is actually being reported to be associated with other autoimmune disease. But I can't remember them now, but yeah, we do see that trend.
And one clarification, if I understand it correctly, you said DR1502 as the, you call it susceptibility allele in your cohort for SLE?
Yeah, okay, I cannot show the slide. Yeah, the susceptible ones is the DR2 and DR3.
Okay, so if we go by the, these are very commonly seen in the Asian population. So do you want to call it association, or you want to call it susceptibility? How do you go about, it's an association, or you say that they are actually causing susceptibility?
Yeah, good question. So again, we are like comparing between healthy control and the case. So in this case, we will prefer to call this as a susceptibility because it's the way the project is designed. But yeah, maybe you will have other comments on that, like how do we call that in the sense of clinical studies? Yeah.
So how do you see the HLA association being, how do you think it can be translationally used? Because it doesn't figure out, it doesn't figure in the list of the, the EULAR classification. It doesn't figure out the HLA susceptibility for. So how do you think this knowledge can be used in the clinics?
Yeah, very good question. Yeah, so again, same as all the case control disease study, carrying a particular allele doesn't make you 100% having the disease itself. So it will only like increasing the risk of the odd ratio. So we have, in this case, maybe our highest odd ratio is around 3 to 4-fold, but then doesn't mean that the people who carry that will be the disease itself. But then I guess what we can use in this case is that, for example, we have some gray area cases that we are not sure whether they are SLE or not, and screening their HLA might help with the proper diagnosis. Yeah.
From Municipal Public Health, Thailand. So thank you for update the research questions that we work together.
Yeah.
And since you established the Thai HLA imputation database, how can Thai researcher or others access to that database and use it for other research?
Yes. Yeah, thank you for the question. So again, you are part of our consortium as well. So I guess that is something to be discussed because this is freshly out, the Thai reference. So I guess that you as a data provider, we will give you a copy of that reference that can be used. And then as a consortium activity, we will want to perform a validation of the reference as well. And the other plan that we have that I want to discuss with you maybe after this meeting is that we have this online imputation system that you don't actually need to share your reference around in case of any ethical problem or anything. And then user just upload their data to a web base, and then the result will be returned to the user, and then no sharing of reference file at all. So that is something that we need to discuss about. Yeah.
Thank you.
Thank Thank you very much for the nice discussion. So thank you for all speakers and the excellent presentations to our audience for the engaging discussion. We hope these sessions have provided insights into the HLA genomic and its clinical application.